Chapter 12, Section 3, Active Reading Review. Number 1, state the central dogma of biology. I think I referenced this in the notes. DNA codes for RNA, which guides the synthesis of proteins. Number 2, differentiate between transcription and translation. Transcription is the synthesis of mRNA from DNA. So when you go from DNA to RNA, mRNA, that is known as transcription. Translation is the synthesis of a protein from mRNA. So the translation is when we utilize the codon charts. When you look up the chart for the amino acids from the mRNA strand dealing with the ribosomes, that is known as translation, because think about it, we're essentially translating the mRNA codons into amino acids, which are then put together to create a polypeptide to essentially create a protein molecule. All right. Number 3, discuss the ways in which eukaryotic pre-mRNA is processed before it reaches the ribosomes. The introns are removed. A protective cap is added to the 5' end, and a poly A tail is added to the 3' end. Number 4, summarize the synthesis of mRNA. Remember, mRNA is messenger RNA, and we get that through transcription from DNA. The DNA is unzipped, and RNA polymerase binds to the template strand of the DNA and moves in the 3' to 5' direction. The mRNA transcript is manufactured in the 5' to 3' direction, and uracil, U, is incorporated instead of thymine, T. And number 5, determine the protein sequence that would be coded for by reading the following mRNA sequence from left to right. So this is where you would open up or look at your codon chart. There's also a codon wheel, which some people find it easier to utilize the wheel. And it's basically like, think of it like a bullseye or a target, where you will start in the middle and work your way out based on the first base, second base, third base of the codon. So to break this down, you would look up UCU, then you would hopefully find serine. Then you would look up CUG, leucine, GCU, alanine, AUC, isoleucine, AGC, serine. All right. So again, you'll utilize your codon chart or codon wheel, whatever. Stop.
Who are you talking to?
Just wait. You would use, sorry about that. You would use your codon wheel or chart to look up those codons in 3s, and hopefully you get the same amino acid sequence as we did here. All right, that's it for me, Mr. Case, signing off.